Hi folks, welcome to my channel, The Average Dermadoc. Let's begin the show. Toll-like receptors are a subfamily of pattern recognition receptors which recognizes pathogen-associated molecular patterns, damage-associated molecular patterns and activates specific signaling pathways. These pathways leads to activation of innate immune response mainly. These pattern recognition receptors are key players for innate immunity. It will also prime subsequent adaptive immune response till the pathogen is eliminated. Coming to classification of pattern recognition receptors, mainly based on location. One is cytoplasmic, which means these pattern recognition receptors will lie within the cytoplasm of a cell. Examples: node-like receptors. Node means nucleotide oligomerization domain. It will function as signaling pathways, which leads to activation of signaling pathways. One is RIP2, which activates nuclear factor kappa beta. Whenever you hear this word nuclear factor kappa beta, it will lead to production of pro-inflammatory cytokines. Another pathway, CORD9, which activates MAP kinase AP1. Upon recognition of pathogen-associated molecular patterns, what are those which is identified by this node-like receptors? Node 1 identifies gram-negative bacteria. N for node, N for negative, gram-negative bacteria. Node 2, along with gram-negative bacteria, it will also identify gram-positive bacteria and activates innate immunity via these two signaling pathways. Node 1 and Node 2 acts via these two signaling pathways. Next, RIG1 like receptors. These receptors acts via MAVS, which leads to activation of nuclear factor kappa beta. What it will lead to? Production of pro-inflammatory cytokines. And IRF3, which means interferon regulatory factor 3. It will lead to formation of interferons, as the name indicates. Next, upon what pattern RIG will get activated? R for RIG, R for RNA. RNA viruses will stimulate RIG1 like receptors, will lead to activation of this pathway, ultimately leading to production of interferons. We all know interferons will act against viruses. DNA sensors within the cytoplasm, AIM2 like receptors, which acts via inflammasome. Another receptor, NLRP3, also acts via inflammasome. This NLRP is nothing but a type of node-like receptor. Until now we have seen node 1, node 2, NLRP3. All these comes under node-like receptors. C-gas receptor acts via STING pathway, leading to activation of nuclear factor kappa beta and interferon 3. Nuclear factor kappa beta will form pro-inflammatory cytokines, IRF3 will form interferons. Coming to transmembrane receptors. Here our topic of interest, toll-like receptor, is a transmembrane receptor. It is of two types, again based on location. Surface, which is lying on plasma membrane. Endosomal, which is lying on endosomal membrane. What is meant by transmembrane? Transmembrane means which crosses the membrane. What toll-like receptors will do upon activation by specific pattern recognition molecules? It will act via mid-88 pathway. Mainly, all the toll-like receptors will act via mid-88 pathway, leading to activation of nuclear factor kappa beta. I already told you, it will lead to production of pro-inflammatory cytokines as an outcome ultimately. We will see what are those examples. TNF alpha, interleukin 1 beta and interleukin 6. Except one toll-like receptor, that is toll-like receptor 3, which acts via TRIF pathway, which leads to activation of IRF3. IRF3, toll-like receptor 3, which leads to formation of interferons, as I already told you. Mainly type 1 interferons, that is interferon alpha and interferon beta. These two pathways are for toll-like receptors. Based on location, again toll-like receptors are of two types, surface and endosomal. Surface means which crosses the plasma membrane and extends to outside of the cell. Endosomal means which crosses only endosomal membrane and lies inside the cell completely. C-type lectin receptors. Examples, dectin-1 and mannose receptor. Dectin-1 acts via signaling. Pathway is Syk kinase, leads to activation of CORD9. Until now, we have seen the function, signaling function of pattern recognition receptors. Next, endocytic function. That is nothing but phagocytosis. Antigen will be taken into the cell and then transferred to phagolysosome, ultimately leading to phagocytosis of the pathogen. What are the receptors acting via this function are mannose receptor, which is a C-type lectin receptor, and scavenger receptor. Where is their location? Mannose receptor, scavenger receptor, dectin receptor, toll-like receptor. Their location is transmembrane. Next, soluble protein secreted by the liver into the bloodstream. This soluble protein will act as a opsonin. Example is mannose binding lectin. Here this CTLR means not control button, it is C-type lectin receptor only. Total 3 C-type lectin receptors. Dectin-1, which will function as signaling pathways. Mannose receptor, which will function as endocytic, leading to phagocytosis. Mannose binding lectin receptor, which is a soluble protein secreted and acts as a opsonin, which activates classical complement pathway mediated phagocytosis. Just it will act like a opsonin. These pattern recognition receptors, along with toll-like receptor, also involved in the pathogenesis of various immune-mediated inflammatory diseases. If they identify self-DNA as non-self and triggers immune response, they will help in the protection against pathogens by recognizing pathogen-associated molecular patterns, and they are also involved in many immune-mediated inflammatory skin diseases also. Coming to our proper topic, toll-like receptors. This word toll in German means fantastic. Already we know it is a type of pattern recognition receptor. It is similar to toll receptor in Drosophila species. That's why it got its name toll-like receptor. Already toll receptor is there in Drosophila. What is Drosophila? It is a fruit fly which has only just 4 chromosomes. Their genetic composition is similar to that of humans. That's why they are utilized extensively in genetic research studies. Toll-like receptors are transmembrane receptors. Already we have seen this. Two types, surface and endosomal. They are present in immune cells like monocytes, macrophages, dendritic, B, T and natural killer cells. They are not only present in innate immune cells but also in non-immune cells like fibroblasts and keratinocytes. We will see toll-like receptors individually one by one. There are total 10 toll-like receptors in humans. Coming to toll-like receptor 1 and 2, it will recognize pathogen-associated molecular pattern, that is triacyl lipopeptides, which will be in bacteria. Next, toll-like receptor 2. I have typed here instead of 2 directly Z for zymosan, which is a component of yeast cell wall, nothing but fungi, which protects us against, which develops innate immunity against fungi. we will say di for 2. It recognizes diacyl lipopeptides. MALP2 is specific diacyl lipopeptide for mycoplasma. So it also works against mycoplasma. Another pathogen starting with M, Mycobacteria species, which contains lipoarabinomannan, Mycobacterium leprae, Mycobacterium TB. Both will be taken care of this toll-like receptor 2. Next, peptidoglycan. In gram-positive bacteria, instead of plus symbol, I have used directly T here, gram-positive, because it is identified by toll-like receptor 2. T for 2, T for gram-positive bacteria. Total 4. Zymosan in fungi, diacyl lipopeptides for mycoplasma, lipoarabinomannan for mycobacteria species, peptidoglycan for gram-positive bacteria. Toll-like receptor 3. I have already told, it is the only toll-like receptor which acts via TRIF pathway, which leads to IRF3 and then ultimately interferons, which acts against virus. So it has to protect us against virus only. Within the virus, it will identify double-stranded DNA and RNA. Remember, like this, toll-like receptor 3 will identify 3-letter word DNA and RNA. We will see examples of double-stranded DNA viruses. Herpes simplex virus, herpes zoster virus, human papilloma virus, molluscum contagiosum virus. So, if we get a question on herpes genitalis, its causative organism is HSV. Now we know what is the genetic material, double-stranded DNA. Like that, we have to derive some points from every question, which we can apply on other questions also. Here, EN means endosomal toll-like receptor. Next, toll-like receptor 4. It recognizes lipopolysaccharides of gram-negative bacteria. If you have already seen my video on lasers, there I have told for ND-YAG laser, convert that G to 4. Now here I am doing the reverse, converting this 4 to G. G for gram. I am extending this 4 to get a negative symbol. Gram-negative bacteria. Toll-like receptor 4 recognizes gram-negative bacteria. Coming to toll-like receptor 5. F for 5, F for flagellated bacteria. Toll-like receptor 6 and 2 recognizes diacyl lipopeptides. Works against mycoplasma. Toll-like receptor 7, S for 7, S for single-stranded RNA. Same for 8 also, single-stranded RNA, which identifies viruses. Example of single-stranded RNA virus, HIV-1 virus for toll-like receptor 7. 8 also same. The specialty of toll-like receptor 7 is it has a drug which acts as an agonist for it, that is imiquimod, used for genital warts and certain skin cancers. Next, toll-like receptor 9. This numerical 9 I have purposefully written it in reverse to resemble P. P for protozoa, mainly Plasmodium, as it recognizes hemojoin. This toll-like receptor 9 also recognizes DNA with cytosine phosphate guanine dinucleotide sequences of bacteria and viruses. It will lead to stimulation of B cells and production of autoantibodies also. So, if we use an antagonist which inhibits this toll-like receptor, we can use for treating SLE and psoriasis. Same if we use antagonist for toll-like receptor 2, we can use in treatment of acne. Toll-like receptor 2 along with Propionibacterium acne is responsible in the pathogenesis of acne. So we can control acne like this. If we inhibit toll-like receptors, we can control unnecessary inflammation also. See, I have written here SLE, psoriasis and acne. Here this SLE and psoriasis are immune-mediated inflammatory disease. Already we have seen that same sentence. Pattern recognition receptors are also involved in pathogenesis of various immune-mediated inflammatory diseases. As we can see here, toll-like receptors are involved in acne, SLE, psoriasis. It is also involved in atopic dermatitis, rosacea as well. Coming to last receptor, toll-like receptor 10, have purposefully written the spelling wrong because natural ligand for this is unknown, but it acts against bacteria. If I keep any heading without writing anything, just with a question mark like this, it means by this time we should be able to write applied aspects on our own. We will see how. First, toll-like receptors will act against many microbes, bacteria, fungi, mycoplasma, mycobacteria, bacteria, viruses. Toll-like receptors, if we inhibit them, we can control unnecessary inflammation. Point number 2. 3, we can use it for cancer therapy and we can use it for certain diseases like genital warts, which are agonist for toll-like receptor 7. Example, imiquimod. These toll-like receptors are also used as an adjuvant for vaccines. This toll-like receptor antagonist will be useful in controlling immune-mediated inflammatory disease like SLE, psoriasis and acne also. 5 points already within this table we are able to derive as applied aspects. My main aim here is to give you a crisp and concised material which you can revise fast just a day before the exam, not to overload you with content. If you learn just this table, it's more than enough for toll-like receptors. This is the picture I have taken from Robbins. Just to see this in your exam, you have to be in a position to draw this picture and write toll-like receptor. What I told about toll-like receptor, it is 2 types. First, surface receptor on plasma membrane, endosomal receptor on endosomal membrane. See here, it is recognizing bacterial products outside the cell. That means the active domain. Transmembrane receptor will have 2 domains. One is intra, one is endodomain, one is ectodomain. Here the active domain is ectodomain. As you can see, it is recognizing bacterial products outside the cell. Here you can see it is recognizing genetic material inside the endosome. So endodomain is here active. Ectodomain is within the cytoplasm. Here ectodomain is outside the cell. Here you should get a doubt. This endosome is within the cell only, inside the cytoplasm. Why we are not calling this toll-like receptor as cytosolic, like NOD-like receptor and RIG-like receptor? We are calling them as cytosolic because this active domain is recognizing and functioning only inside the endosome. If the nucleic acid is ingested into the endosome, then only this active domain will work within the endosome only, not within the cytoplasm. But here in case of NOD-like receptor, RIG-like receptor, if the pathogen is inside the cytoplasm, it will recognize and works. Here this ectodomain, which is outside the cell, will have leucine-rich repeats for surface toll-like receptors only. This endosomal receptor ectodomain is within the cytoplasm only, but it is not the active domain. Here you can see lectin, nothing but C-type lectin receptor. Here it is crossing the membrane. That means it is a transmembrane receptor. What is the example of C-type lectin transmembrane receptor? Here it is identifying polysaccharide. It is nothing but mannose receptor, which is recognizing polysaccharide, mannose. I already told you R for RIG, R for RNA. Here you can see clearly it is recognizing viral RNA. Remember the channel name. Don't forget to like, share and subscribe. Thank you. Hasta la vista. До свидания.